Hopefully my presentation will reflect some of the aspects of clinical translation. Most of it's going to be actually preclinical with, using drug discovery, developing drugs to move into the clinic. I have got some examples where PerkinElmer are trying to make steps into the clinic as well, using things like image-guided surgery, looking at aspects of diagnostics. So I'm going to show some examples of that. So I've kept it very, very broad. I'm going to start off by explaining the aspects of optical imaging, since this is an educational session, and then move in to show you some models. So just to start off by pointing out the obvious, which you probably already know, if you look at the absorption in an animal, the absorption of hemoglobin when light is passing through tissue, you find that really it's not until you get to about 650 nanometers you've got a clear optical window. And this is what has been, I've worked for what was Xenogen, then Caliper, now PerkinElmer for the last 15 years doing optical imaging. And when we first started out, we were very fortunate to be using Firefly Luciferase for most of our models, which had a very, very broad spectrum and goes right up to about 800 nanometers in its wavelength. And so because of that, that was fortuitous, because it meant all of the models we were developing had very good penetration. With fluorescent imaging, however, it was more problematic, because a lot of the initial fluorochromes that were being developed were either very blue, or if they were redshifted, became very dull, and that caused a lot of issues. And the best way of showing this is, as you can see this image on the left-hand side, if you look at two light sources, you have green light and red light. Best way to explain it, and the reason we got these little devices is because when we would go out in the field, people would always say to us, we want to image GFP. And GFP is maybe a fantastic reporter for in vitro. When it comes to in vivo imaging, deep imaging, it just doesn't work effectively. And so if you put a GFP source behind your hand, you can see that basically none of the light, or virtually none of the light, gets through. Whereas with red light and far red light, it comes straight through, and that's a big difference. It's just a fundamental physical difference. So most of the imaging platforms, and I've tried to make this so it's not a sales pitch, so I've made generic images for you, just to show you how most of the imaging platforms work. They're essentially dark boxes. When you're dealing with bioluminescence or chemiluminescence, you have to scavenge for every photon to be able to detect a signal through an animal. And so that means you have to have these light-tight boxes with very, very sensitive CCD cameras. Most of these cameras were developed for astronomy, for looking at distant galaxies. So when you turn them upside down and put them above an animal, it means you can detect single photons. Or you can also have fluorescent imaging platforms. The major difference between these is with a fluorescence dedicated imaging platform, you don't need such a cooled system. It can be cooled to minus 40 as opposed to minus 90 or below. This allows you to get more sensitivity. It also allows you to detect further into the red as well. So these are the two real configurations that people have when they're developing optical imaging platforms that offer small animal imaging. So the basic methodology is that you take a bioluminescent or fluorescent or chemiluminescent, any optical reporter you want, and then introduce it either into a cell line. So a lot of our customers obviously are tracking cancer models or stem cells or infectious disease agents in the animal. And so what they do is they engineer the cell line and then place the optically engineered cell line into the animal to monitor and track it. The alternative is, of course, that you can take these transgenes and introduce them directly into the animal to make a transgenic animal, either by using pronuclear microinjection, or you can make somatotransgenics, where you introduce the gene after birth. You then take the animal with its optical source internalized and place this into the imaging platform. The standard method is simply just then to capture an image of the animal, which is a photographic image, and then the optical image is collected over time. If it's bioluminescence, if it's something like a bacterial infection, which can be incredibly weak, you may be scavenging photons up to 5 minutes. When it's fluorescent imaging, it can be a split second. And a lot of the time now people use composite imaging, where they'll take, for example, a cancer, and then they'll identify a target on the surface, the cancer would be labeled with bioluminescence, the targeting system would be using fluorescence. So you can combine different optical imaging modalities into one system. Of course, the other big advantage of any imaging modality is the fact that you can reduce animal usage. So originally I came from Europe, where it's a much bigger issue to try and reduce animal usage. But here now in the US also, it's become a large issue, and the 3Rs has really promoted this type of technology where you can do this longitudinal imaging. Instead of sacrificing animals, simply image the same animal over and over again. And of course, the big advantage of that as well is that if you do have, for example, you're doing an efficacy study for a cancer, you can treat the animal, you can see diminution of light, which is indicative of the cancer dying and the drug being efficacious, but you can see then whether the cancer comes back, because the light will come back with the cancer, same with infectious disease. Another aspect of the imaging technology is now to use composite imaging. So optical imaging is very similar to PET imaging. It tends to give you a very sensitive image, but it's very broad and it has very low resolution. So now what people are doing is they're doing combined imaging, multimodality imaging. Well, they image the animal initially in an optical imaging system, and then they may combine it with either CT or MRI, so they get good anatomical registration. And that shows an example here, and we build these little cassettes that you can move from one imaging platform to another. Or, of course, this is just one little bit of a sales pitch. This is our all singing, all dancing Spectrum CT. This imaging platform will do bioluminescence, fluorescence, chemiluminescence, phosphorescence, Cerenkov imaging, plus it has a flat panel CT, which is so fast that we had to slow it down. When we first developed this CT system, one of our mad physicists saw how fast he could take an image, and he could take an image down to a split second, down to 1 second, but the centrifugal force on the stage was so high that the mouse would fly off the side. So we had to slow it down to 3.5 seconds to be able to do an image. But it's incredibly fast. The optical is very fast and the CT is very fast. So you can generate an image, a full 3D image, in as little as a few minutes. So I'm going to start off by giving you some examples. A lot of the examples that we've generated are in infectious disease simply because it's a much easier model to run fast and to change a lot of the different parameters. So a lot of the images I'm going to show you are in infectious disease, and I'm going to move on to show you some oncology and then some inflammation studies that have been done. Most of the bacterial studies that have been developed have been using bacterial luciferase. The advantage of bacterial luciferase, you don't need any substrate. You can engineer virtually any bacteria you can think of simply by introducing a full, what's called a LOTS cassette, which is a 5-gene operon that can be introduced into the bacteria and make them constitutively glow. And so you don't need anything else. The bacteria simply glow once you've engineered them. So very simple imaging. Start off very simple just to show you how people use the technology predominantly in pharmaceutical companies, but also just to see if a bug is pathogenic or not. This just shows you an example of with strep pneumoniae, where it's an IP injection with 10 to the 5 bacteria, and then you can see proliferation of the bacteria over time. So you're just doing longitudinal imaging, same mouse, just watching expansion of the disease. And one of the nice things about using this is it's very quantitative. So you can see here that you're getting this from blue, which is a very dull signal, up to when you've got this intense red, which is obviously a high number of photons, which is the same as having a high number of bacteria. There's a very good correlation coefficient between, as the pathogen increases, the light increases, of course. So this means you can predict not only if the animal will die, but to what hour the animal will die. And the reason we obviously want to do this is we can do rescue studies, but also you don't have to run the animal studies for death as an endpoint. So it means you can tell immediately if the animal is going to be overwhelmed, and you can sacrifice it if you need to. This again is a typical type of study, an efficacious study for different drugs, just testing a vehicle against daptomycin and ceftriaxone. And you can see here that with the vehicle, if the animal starts developing light, it dies. So you can see that it's actually very indicative of what you would see. The animal gets the infection. If you don't treat it, it's going to end up dying. Whereas here you can see at 2 hours they've treated, and in this case these two animals survive. So it's a way of being able to rescue the animals, which also can then tell you if you've got a splice or SNP variant or something with the genetics or some adverse effect from the antibacterial or anti-cancer drug, whatever you're testing. As I said, we also like trying to combine optical imaging with anatomical imaging. So we sell a platform which has an X-ray unit built into it as well. This is a study using Salmonella typhimurium in a gastrointestinal tract infection. You can simply see here that we've taken Salmonella and we've engineered it not only with bioluminescence but also with cherry. And you can see that it actually changes the bacteria to a red color. That's how much fluorescent protein is being produced in this bacteria. And in this case, you can see you can image by the bioluminescence, fluorescence, and then you can also get, obviously, anatomical registration. What we did here, we simply took Salmonella typhimurium, mixed them with peanut butter, which the animals like eating, and then laced the peanut butter with barium sulfate so we could see the X-ray image as well. So that way you can do this co-registration very easily, just in one platform. And then you can move on. You can actually, in our 3D imaging platforms, which are combined with CT, you can start getting this full anatomical registration and see exactly where the pathogen is proliferating, in this case in the small intestine of the gastrointestinal tract. This is using Salmonella typhimurium again. And what we've done recently is we've been working with Imperial College in London, and they have one of our spectrum CTs. And so what we've done is we've started making these what are called 4D movies, where we take 3D images and overlap them so that we can see longitudinal progression or diminution of the light. We're doing a drug study. And the next image is just showing you a very simplistic version. The original one was published on the BBC and has 12 images. This is just 3 images because I don't want to keep you here all day. But it just shows expansion of the bacteria over time moving through the gastrointestinal tract. This is actually Citrobacter rodentium, which causes inflammatory bowel disease in a mouse model. So I'll just show you that one more time. So you can see it starts off quite a weak signal, moving from the small intestine into the large intestine when it starts to proliferate more rapidly and then bloom to give this full-blown inflammatory effect. But if you're looking at infectious disease, I'd say most of what I've shown you here is looking at antibacterial, or looking at bacteria, but you can also look at viruses, and you can even look at infectious agents that normally you couldn't monitor directly. For example, this was a study we published with a Nobel laureate, Stan Prusner. When I was in the UK, I ate too many beef burgers during my postdoc, and that was during the height of the BSE crisis, so I was convinced I would get Creutzfeldt-Jakob disease. So when I moved to the Bay Area and Stan Prusner's down the road, we ran this study with him to see if we could monitor prion damage in the brain of transgenic animals. So this is a transgenic animal developed by pronuclear microinjection, where every cell is labeled with luciferase, but it's only expressed in the astrocytes because it's using the GFAP promoter. So when you get damage to the astrocytes, neuroinflammation, you can see light from the animal's brain, as you can see here. So this shows you damage to the brain. And Stan's actually now gone on to develop these models, not only for prion damage, but also monitoring Alzheimer's as well. A lot of prions also form amyloid plaque type damage as well, the same as you see with Alzheimer's. And then this just shows you where we made a composite image, where we're using a 7 Tesla MRI, which is down at China Basin at UCSF, and we've simply taken these very crude, these 10 slices with the bioluminescence throughout the brain and made these composite MRI optical images. Again, another example of transgenics. This, again, I'm very fortunate to work with a lot of really smart academics. This is work that's been done by a guy called Eldad Hod, who's at Columbia University. And what he's done here is he was showing that if you transfuse animals with old or fresh blood, you can show that you get a really pronounced inflammatory response by using old blood. So you can store blood up to 42 days, as I'm sure a lot of you know, and then that blood can be used, obviously, to transfuse. So it's stored for long periods of time in case there's a major accident. But having fresh blood is actually much healthier for you. You don't get breakdown of the red blood cells. And you can see here, this is a transgenic animal which is very sensitive to inflammation. It's the SAA, serum amyloid A promoter, that's firing this luciferase construct. And you can see here that if we use fresh blood, there's virtually no inflammation whatsoever. But by using blood that's been stored for just 4 days, you can see you get this massive inflammation. Not only that, we've just submitted 2 publications where if you take bacteria, mostly enterics, gram-negative, such as E. coli or Salmonella, if you cause an infection in these animals, you find that you get almost 100% mortality from these animals using stored blood. And that's because of the iron allows the bacteria to proliferate. So if you can imagine, if you're involved in a car accident and you get part of a fender or something jammed into your leg, you're losing blood. They try and stabilize your blood pressure by giving you epinephrine, which is the growth factor for bacteria. They then give you used blood, and if you've got contamination by bacteria in your bloodstream already, it could lead to higher mortality. So it's actually doing these types of studies allow you then to try and figure out which is the best treatment to have. We also do a lot of surgically relevant models as well. This is work that's been done by Lloyd Miller, who's now at Johns Hopkins, was at UCLA. This is a biofilm model. Again, it's looking at bacteria. We've put a Staph aureus at about 10 to the 3, 10 to the 4 onto the end of this pin. This is a titanium pin which has been placed into the femur of the animal, and you can then see proliferation of the bacteria on the end of this pin. This is very similar to what you would see again if you had a hip replacement and it had gone wrong and you got an infection, a post-operative infection, which happens here in the US about 3 to 5% of the time. It always jams up at least one time during my presentation. Got too much junk running on it. Okay, so again, when we show a lot of these slides to people, they, especially in Europe, they get horrified because they think we're using huge numbers of animals. But what you can see here, this is a massive amount of data. We've taken again, a transgenic animal which is expressing, in this case, GFP, because it's close to the surface, so GFP can be seen close to the surface. It's a GFP transgenic animal with a neutrophil population is labeled with GFP. So you can see tropism of the neutrophils to an inflamed site. And the bacteria are on the end of this pin producing bioluminescence. So what you can see here is that you can monitor progression of this biofilm on the end of this pin, and you can also see the tropism and then quiescence of the neutrophil population attracted to this inflamed site. And you can monitor these 2 events simultaneously. But what you can see here is all this data is generated from 3 animals. Each one of these is an animal. And the reason I put this on here is to show that the disease profile is dramatically different between these 3 animals. This is what's most commonly seen, but you get a lot of outliers such as this, where the signal goes up, goes down, then disappears. It's all over the place. And this is what you normally see with infections in the clinic, is that no 2 individuals give you the same infection. So because you can do longitudinal imaging, you can track this in real time and get a real indication of how the disease is progressing. We also, because we have these flat panel CTs at the same time, we've got this cause and effect. A lot of the time, of course, what happens when you have, for example, a hip replacement, it gets infected, then the bone gets destroyed by the neutrophils and macrophage invading the bone to try and eradicate the bacteria, and then it causes this crumbling of the bone, this lower density, and the hip can collapse. And this is what you see in this model here, is as the bacteria proliferate, you can see these holes developing in the bone, this demineralization, and then the leg will snap. And again, just simply showing you that we make these composite images so we can do a close-up image. All those images that I showed you on the CT were all done in living animals, so we can simply take a high-resolution close-up CT imaging, show the damage to the bone, come back out, and start seeing proliferation of the bacteria on the pin and also the neutrophil signal at the same time. So this is where we're doing now more clinical translation. Again, I'm fortunate enough to work with a group from Groningen University, so and now working with a group from Johns Hopkins as well. A group from Groningen do a lot of image-guided surgery. You probably know Professor Go Van Dam works with Vasilis Nather-Christos in developing image-guided surgery systems. And what we did in this particular study, which has just got accepted into Nature Communications, is we were taking vancomycin, which targets gram-positive bacteria, and we've labeled it with a near-infrared dye. And by labeling it with a near-infrared dye, it allows us then to act as a theragnostic. So it's normally a micro-dosing level, so it doesn't really affect the bacteria, but it binds very strongly to the gram-positive cell wall and allows us to label the bacteria. So you can see here we've taken different gram-positive and gram-negative bacteria, and this is we've genetically engineered them all. This is from our stock. We sell these as part numbers. So you can see here we've got Pseudomonas, E. coli, Strep pyogenes, and then different strains of Staph aureus, and then including a methicillin-resistant Staph aureus and then a Staph epidermidis. This is the bioluminescence from the strains, and then we've simply mixed them with this vancomycin and washed them, and you can see that it doesn't bind to gram-negatives hardly at all, because of course the level of peptidoglycan is much lower, whereas with the gram-positives, Strep pyogenes is not great, but the gram-positives, the Staph aureus and the Staph epi, you get very good labeling with this vancomycin. So what we then can do is we can do targeting in the animal. We've taken an E. coli and a Staph aureus, and then we've injected the animals with this vancomycin labeled with a near-infrared dye, and you can see that it targets the gram-positive bacteria at the disease site or the wound site. And the idea behind this is, of course, we can move from preclinical models to make clinical targeting agents. So Go worked at a hospital, so he had access to lots of dead parts of people. This is the cadaver leg from an amputee, and it shows a plate that's been screwed into the leg, which has a Staph epidermidis infection, and you can see that you're able then to see targeting of this vancomycin to the bacteria, and they can be imaged then directly. This is imaged on the IVIS system. This is imaging in an image-guided system. So the idea is that when you're undergoing a procedure in a hospital, if you have an infection, you can then go in and remount or debride the material that's infected and leaving the uninfected material behind, so you can guide yourself doing the surgery. It's already been shown to work very efficiently with oncology for things like ovarian cancer. It's already been used in the clinic, but now it's going to be used also for infectious diseases as well. And we do all these confirmatory studies as well. We have, again, PerkinElmer is a very large company, has a lot of different imaging platforms, so we have these called nuance cameras, which allow us to unmix microscopically the fluorescent signature at the same time. So what we've done here is we've taken the thigh material from the thigh that was infected with Staph aureus and then the E. coli, and you can see here, this is on the IVIS system, doing a close-up image. Then microscopically, we can even see the gram-positive cocci that are labeled with the near-infrared dye, whereas the gram-negatives are completely unlabeled. So moving on, I'm just going to quickly now show you some examples in oncology, which is our biggest selling area. The only thing I really want to emphasize here is the fact that with cancer, it's incredibly sensitive. We can actually, my colleague J.B. Kim published a paper back in 2010 showing you can detect a single cancer cell subcutaneously in an animal by using optical imaging. So we have exquisite sensitivity. We can detect metastatic lesions down to about 100 cells. And as you know, there's about 2,000 imaging platforms worldwide. So it's a very well accepted technology, and about 60 to 70% of those are in oncology. Also, by using combined imaging, we can use it as a CT, or we can use it as an X-ray overlay. So you can see then if you've got occurrence of metastases in the bone, you can get this spatial definition as well. And then, of course, exactly the same as we did with infectious disease studies, we can do longitudinal imaging, watch progression of the cancer over time. We can watch progression of the damage in the bone using the CT, and then generate these composite images, which can all be done in one imaging platform. The other nice thing about the Spectrum CT is it has the ability to image 2 animals at a time for both optical and CT. So what we're doing at the moment is we're generating a 4D movie by having a drug study where we have a treated and a control animal. And then the nice thing about with pharmaceutical companies, you have to give them what's called an elevator speech. You have about 30 seconds to sell them on the technology. So you can show them this type of image where you can show one animal which has the drug showing efficacy, the other animal which is untreated, you show expansion of the cancer. So it's a very nice way of very quickly showing how your technology can be used for drug discovery. And again, we just make these images where we can show targeting. We combine optical, bioluminescence with fluorescence. This is a lung cancer model showing the cancer in the lungs of the animal, and then targeting with a near-infrared dye. showing that we get very nice co-registration. So this could be, for example, a therapeutic antibody that's labeled with a near-infrared dye, showing the targeting. Also, a lot of our customers use this technology, combining bioluminescence fluorescence, even two-dimensionally. This is showing movement of cancer through the lymphatic system. You can see that there's two sites where the cancer is, but if you put a near-infrared dye into the animal, you can even use ICG for this, as you can see here. You can see flow in the lymphatic system from this point to this point, showing how the cancer can spread through the lymphatic system. Don't want to dwell on this too much, but we've now also got, we're lucky again to work with a lot of different of our customers and then develop little spin-out companies. This is work by Elena Dubovskaya, who's from the EPFL, and she has developed a lot of different cleavable luciferins. Obviously, Promega develops these as well. She's come up with a new chemistry where you can then start modifying the luciferin so they're activatable due to a certain enzymatic activity. And the nice thing about that is then you just engineer your animals once. So if you're looking, say, for example, at cancer, you can then say, well, okay, is my cancer undergoing apoptosis? You can look at caspase activity by using a caspase cleavable luciferin, which I think I have an example of here. Actually, could you, let me move on, that's okay. Let me show you an example here. So this is work that was published using Promega's caspase cleavable luciferins. And it shows you where you've got a model where you have standard luciferin and then activatable luciferin with this DEVD caspase cleavable site. If you look at these two models, you can see with standard luciferin, even if you have cells that obviously aren't undergoing apoptosis, you can't distinguish this group from this group which are undergoing apoptosis. There's no difference between these two sets of animals. But if you use cleavable luciferin, you can see that there's virtually no light unless the cancer has been activated to undergo apoptosis, whereas here with the cleavable luciferin and activation, you can see this activity. So the nice thing about using these cleavable luciferins is, again, you can monitor 101 different enzymatic activities and pathways without having to re-engineer your animal model. So we see these as being very popular for the future. And again, these have now been, a lot of these publications are now coming out from Elena's lab, where she's using, again, this modified cage luciferins to monitor these for a variety of different diseases. Finally, just two other sections I was going to quickly show you. So far I've shown you predominantly bioluminescence and fluorescent imaging, but the system can also be used for chemiluminescence. So this was published earlier in the year by my colleague Ning Zhang. This was in Nature Medicine, and it shows that you can monitor chemiluminescence by shifting the wavelengths through quantum dots. So what's been published previously by people like David Pinnaker-Worms is that you can use luminol to monitor activated neutrophils and macrophage. They produce myeloperoxidase, which produces hypochlorous acid, and this is then what produces the flash of light with luminol. But when you inject it into the animal, you find that you get very weak signals. And this blue light, as you saw my little device at the beginning, blue light's even worse than green light. It doesn't pass through the animal. So all that blue light that's produced from the luminol, a flash of light, is trapped in the animal. But we found if you simply just mix quantum dots with the luminol, the light then can pass through because it shifts the wavelength into the near infrared. And so you get very sensitive imaging. And this then can be used even for dual imaging for things like oncology studies. This again was from the same paper. It shows here where we've got a mouse with metastatic cancer being imaged by bioluminescence. You can see where the sites of these cancers are. And then injecting with luminol with the quantum dots, you can then see the inflammation associated with these cancers as well, and also some additional inflammation in the lungs. And then, of course, we can also do all this three-dimensional imaging and reconstructions. And finally, I wanted to show you Cerenkov imaging, then a little bit of clinical imaging. So Cerenkov imaging is also another modality. If you're looking at PET or SPECT probes, it was found in, I think it was 1959, that Cerenkov, a Nobel laureate from Russia, showed that when you look at a nuclear generator, it glows blue. The reason it glows blue is because the radionuclides produce electrons that can pass faster than the speed of light in water. The speed of light in water is only 3/4 C, so because of this you get this sloughing of the light, and it causes like a photonic boom that produces this blue light. So that means, and this is work from Stanford, if you look at either PET or SPECT or PET probes, you can then monitor using an optical imaging system, these radionuclides, these PET or SPECT probes as well. It's obviously not as sensitive as using a PET or SPECT imager, but it's great for high throughput screening because you can image up to 10 animals at a time in an optical imaging system, pick the animals you're interested in, and then do full three-dimensional imaging in your PET or SPECT imager. And finally, because I'm running out of time, clinical diagnostics. Again, this is work by Go Van Dam. This is just showing you how we're also moving into clinical diagnostics using the imaging platforms. This is a patient that has cancer in the lymph nodes, and it's cancer in their groin. So what they've done is injected the patient with pattern blue, which is the standard. But also pattern blue, it turns out, like most, or a lot of fluorescent or chemiluminescence or colorimetric dyes, they also have a fluorescent signature as well, a near-infrared signature. So you can excite pattern blue to give you a near-infrared signature. So what they've done here is they've taken these lymph nodes, put them in the IVUS system, and then you can see here, this is where the cancer is, here and here. You can see it's blue, and you can also then see the fluorescence. So in this case, there's no advantage. You might as well look at it by eye. But in this example here, you can see these are two other lymph nodes that goes resected from this patient. And when you look at them, they appear negative by visual examination. But if you put them in the IVUS system, you can see they're actually positive for cancer. So we see this as being a big market for the future for not only things like image-guided surgery, but also for doing clinical diagnostics so you can really diagnose and do personalized medicine for patient samples. And that concludes everything I was going to talk about.